Hello, and thank you for tuning into this presentation, which is an introduction to Glycostem. Glycostem is fully focused on NK cells of three different generations. We have the non-manipulated NK cells, CAR-NK, and TCR-NK. After that very brief introduction, a quick look at our finance overview. Last year, we concluded a $28 million fundraise, and adding to the other fundraises we have done, we are up to $35 million raised both in equity, soft government loans, EU grants, and deal money. We are right now out trying to raise a crossover fund of 40 to 50 million. We are looking at closing that in October of this year, and that is obviously leading up to an IPO in Q2 of next year. Even though we have raised a lot of money and still have a lot of money in our bank account, we are looking at raising more money in order to accelerate our CAR-NK and TCR product portfolio. We obviously also know that we need to do a phase 3 clinical trial once our soon-to-be started pivotal trial with Oncourt in AML has been done. And as in many other biotech companies, we have many ideas for improving product and coming up with new products. At the bottom of this slide, you can see how we have in and out license deals going on. We have 2 unannounced deals in the target area for CAR-NK products, and there are many more to come. But we're also working on outsourcing or out-licensing, products to other companies, and hopefully within too long, there will be more good news in that area. The company Glycostem is from 2007. Over the last 3 years, we have started raising money, which has allowed us to build our own GMP licensed manufacturing cleanroom. It was licensed in June of last year, and that obviously allows us to produce our own NK cells for our own clinical study to be initiated in October. We have also put on board a very experienced management team which cover all the traditional aspects, and we are really ready to get started on treating patients. We have developed 2 types of assets. Asset number 1 is obviously the NK cell therapy, and asset number 2 is the processing manufacturing of these. We are using the umbilical cord blood as our raw material. We separate stem cells CD34+ from that, which we then expand, differentiate, and further expand using our own proprietary medium. We are right now having orphan drug designation in the US and in Europe for AML, and we hope we will achieve another orphan drug designation before the end of this year. And as you know, we have developed CAR-NK products for a little while now and are getting really serious into TCR-NK cells as well. And all of what we do is covered by 10 patent families, which have given us a very unique sole opportunity with the CD34+ stem cells derived from umbilical cord blood. We all know, and we all thank you for... sorry. You're still okay. There, I hope.
That's okay.
Do we have time to start all over again? I promise it's the last time we do it.
We have 45 minutes. I'm all yours, TJ.
Oh, you will have a long lunch break then.
So go ahead on back to the beginning there.
Okay, hang on. My mobile phone cancel. Okay, so I count to 5 and I start. Is that okay?
That is correct. I'm going to go on mute now.
Thank you. Thank you for tuning in to this presentation about Glycostem and the development work we are doing. At Glycostem, we are 100% focused on NK cells. We are working on first generation being non-manipulated NK cells, second generation being CAR-NK, and third generation being TCR-NK. If we look at Glycostem from a financial overview, we have, since the start of our company, raised about $40 million US, and last year alone, we raised about 28 million. That is enough to see through our upcoming NK cell trial in AML. But we have many other ideas for how we can spend our money. So we would like to raise more money for the CAR-NK product acceleration. We want to get into phase 1 with those products. We know we need to reserve money to do a phase 3 clinical trial. And we also want to be ready to satisfy the market need for our product Oncourt when it is finally approved and ready to be launched in 2024. We are right now finalizing a crossover fundraise of 40 to 50 million US, and that is the lead up to an IPO next year, where we hope to raise about 100 million. As you can see from the bottom of this slide, we have several in and out license deals taking place. These out license deals are focused around CAR-NK, but we are also very heavily into the TCR-NK area. When you look at the company, we got started back at 2007 and have for 13 years been perfecting the understanding of NK cells. We have, over the last 3 years, raised an increasing amount of money, which have seen us moving into state-of-the-art research and development facilities. And we are now getting ready to produce in our own GMP licensed facility NK cells for the upcoming WINK study on non-manipulated NK cells in AML patients. As a company, we have invested heavily in the process of NK cells, making sure it's a closed system. We are introducing an electronic batch record system to make sure that we have a smooth and professionally running clean room setup. We harvest all our raw material from umbilical cord blood. We are looking for the stem cell CD34+, which we expand, differentiate into CD56+, and then further expand. We have orphan drug designation in Europe and in USA for AML, and we hope soon to expand that for other indications. We have 10 different patent families that cover process and product, and we are delighted to say that we have a single opportunity in the market as the only company really focusing on umbilical cord blood derived CD34+ cells. When it comes to NK cells and compare them to CAR T cells, I am sure that the audience is well aware of CAR T cells having blazed the trail for NK cells to follow them and have left ample opportunities to improve. For instance, NK cells have much less neurological toxicities. We know that NK cells are less expensive to produce, and we also know that off the shelf is what patients and doctors want. And with all the autologous CAR T cell investments taking place, that's going to be a little while before there will be any competition there. And happily, it's also good to see how so many academic people are turning around and saying that NK cells will actually be able to kill cancers that CAR T cells will not be able to, because the cancer cells can hide. NK cells are obviously a very important part of the immune system. They were discovered about 45, 50 years ago, and it was really not until Miller in 2005 did his landmark publication that it started hitting the headlines. As mentioned earlier, we will in October initiate a clinical trial. It's a phase 1/2A pivotal trial. We will have investigator meeting in early October, and we will be ready to treat patients in November. The first part of the study is 9 patients in a phase 1 study, followed by 24 patients in a phase 2A study. We are repeating our original phase 1 study because we have moved the production facility from a university hospital to a privately owned clean room, and we are supplying an off-the-shelf product that is frozen upon arrival instead of fresh product. About 18 months is what we expect it will take to recruit 33 patients. We will have 1 year follow-up, which will be followed with about 150 days of regulatory review. After AML, we will move smoothly into the study. We will skip the phase 1. We borrow the data for from the AML study, and that will follow a similar route. Now, as mentioned, it is not our first phase 1. Here is a reminder of the excellent data we had from our first phase 1 in AML patients. It was obviously a safety study, and we are delighted to say that there were no NK cell related side effects. There were some side effects that could be related back to the Cy/Flu preconditioning treatment. On the right-hand side, you can see what the Dutch Cancer Institute told us would be the overall survival of other patients of the same category. And at year 1, you would expect 35 to 40% survival. We had 80. And at year 5, you would expect 5 years survival. And we had 50% up till year 5. Here's a better overview of the phase 1/2A trial design. As you can see, we will do a typical 3+3 study up front, and that will then be followed with 24 patients in the phase 2A part of the study. We have already, at time of recording this, received 4 countries' approval to carry out this study. And by the time you're listening to this, I'm pretty certain that we will have all 5 countries approved our study. So we will have 8 clinical sites. We first precondition with Cy/Flu, then we move over and give 3 doses of Oncord, and that will then be followed up by MRD measurements using both flow cytometry and next generation sequencing. From this slide, you can see that it's not only Europe that we have approached. We have also been in contact with FDA. Our pre-IND submission was met with excellent feedback, and that gives us hope that we can submit in Q1 of 21 our IND and hopefully get approval in Q2 of next year. And if we manage to make all of this happen, then the US approval process should really be aligned with the European process. We are, as you have heard, been active in CAR-NK for some time, and this slide really shows how we did our first animal study. So it's a liver model, and a normal mouse liver is somewhere between 1 and 2 grams. When you treat mice with K562 and treat them with saline, you can see in the first column that the liver weight will increase to 6 to 10. And to make a very long story short, if you look at the last column, you can there see that we almost normalized the liver weight in the mice. We have many good ways of improving the efficacy of our product in mice. So we hope that when we early next year release our next animal study, that you can see that we have normalized some of the mice liver weight. As I said at the beginning, we are proud to be working on 2 different assets. Asset number 1 being the NK cell therapy, and the other one is the processes, the GMP processes. There are all too many examples for how it goes, companies who do not give this proper attention. So this is where we have focused over the last 6 years. We start an 8-step process by having umbilical cord blood, fresh or frozen, supplied to us in a closed bag. We then separate the CD34 plus, put them into static culture for 2 weeks, move them over to Zurich wave reactors for 4 weeks. And during these 2 culturing steps, the cells will be expanded, differentiated and further expanded. We will right now in our large scale system see up to 10,000-fold expansion. And we know from our R&D model that we can get up to 50,000-fold expansion. So after 4 weeks on the wave reactors, the cells will be washed, they will be harvested, put into 25 ml transfusion bags and frozen. We have developed our own proprietary freezing algorithm, which will optimize the survival of NK cells after thawing. Then all the products will go into our biobank where they will stay for 2 weeks until we have received all the sterility and mycoplasma test results back. So this is the way we have set up the system, and at no time point between step 1 and step 8 will the cells be exposed to the atmosphere. And that is what prevents wasteful contamination of the final product. So this was a quick, brief introduction to Glycostem. We are always open for ideas, co-development. We are open for business development discussions on our lead product in Europe and US. And we hope that there has been some new learnings from our presentation today. Thank you very much for your attention.